Welcome, everyone. I am so glad you're all here. Thank you for joining us for the 2025 F.A. Gilfillan Memorial Lecture. I'm Eleanor Feingold. I'm the Dean of the College of Science. Tonight, we're privileged to hear from one of our esteemed colleagues, a scientist whose groundbreaking work has reshaped our understanding of protein science. Those of us who are attending in person tonight, I'm going to rearrange so I can make sure I stay on the mic, especially for the people on Zoom. Those of us who are attending in person tonight are joined by Gilfillan family members, as well as students, relatives, and friends of Elisar Barbar, our Gilfillan Awardee. I'm going to begin with our land acknowledgement. Oregon State University recognizes the impact that its land grant history has had on Indigenous communities in Oregon. We accept responsibility for understanding the continuing impact of that history on these communities. Oregon State is located within the traditional homelands of the Marys River Band of the Kalapuya, who were forcibly removed to reservations in Western Oregon. Today, descendants of these people are part of the Confederated Tribes of Grand Ronde Community of Oregon and the Confederated Tribes of the Siletz Indians, some of whom are part of the Oregon State community today. We deeply value their presence and ongoing contributions to the university and to the College of Science. It is our privilege and our responsibility to support the success of students from Native and Indigenous communities and to support the communities themselves with our work. The Gilfillan Lecture, which is a long-standing tradition in our college, brings us together every year to celebrate outstanding science at OSU in honor of Doc Gilfillan, whose record of extraordinary science has positively impacted generations. Francois Doc Gilfillan served as Dean of the College of Science for nearly 25 years, from 1938 to 1962, and as acting OSU president from 1941 to 42. The Gilfillan Memorial Award honors Doc Gilfillan's life and recognizes his extensive service and contributions to OSU and the field of pharmaceutical chemistry. His contributions are a lasting inspiration to us even in the work we do today. I would like to thank the members of the Gilfillan family who join us virtually and in person today for your family's ongoing investment in science. Through the F.A. Gilfillan Memorial Faculty Award, you're enhancing our efforts to provide innovative teaching for tomorrow's scientists, conduct cutting-edge research, and share impactful scientific discoveries to extend the reach and impact of science. We also celebrate Bob Jensen's and his family's generosity in establishing a fund in memory of his late wife, Nancy, daughter of Doc Gilfillan. This fund will recognize an outstanding College of Science graduate student each year, furthering the Gilfillan's family's enduring impact on science and education at OSU. Through this commitment, their legacy will continue to shape the future of scientific discovery. And now, I'm honored to introduce Elisar Barbar, Department of Biochemistry and Biophysics Head, and recipient of the F.A. Gilfillan Memorial Award for Distinguished Scholarship.
You agree?
I have to say nice things about you before you come up. Plan another 10 minutes. This award recognizes researchers in the college who demonstrate a long and exceptional scientific career with nationally and globally recognized scholarly achievements. Elisar has devoted her career to exploring intrinsically disordered proteins, a unique class of proteins that, unlike traditional proteins, remain flexible rather than adopting a fixed structure. Instead, they remain dynamic, enabling them to perform unique functions within cells. Through NMR spectroscopy and protein biophysics techniques, her research has deepened our understanding of how these proteins interact, with important implications for neurodegenerative diseases and drug development. Over her 2-decade career in the college, she has authored more than 90 refereed papers and reviews and secured nearly $10 million in research funding. She conducted valuable research into the structure of the SARS-CoV-2 nucleocapsid protein during the COVID-19 pandemic. Elisar has also played a key role in strengthening OSU's biophysical research infrastructure for all of our researchers, helping to secure an 800 megahertz nuclear magnetic resonance machine that's elevated the university's standing as a regional leader in the field. Her leadership in science and in her department has fostered a collaborative and inclusive academic environment. She's a strong advocate for inclusive excellence. She's been instrumental in expanding opportunities for women and undergraduate groups in science. Elisar was raised in Beirut, Lebanon, which I think she'll say a little bit more about in her talk, received her undergraduate degree in chemistry at the American University of Beirut amid civil unrest. She decided to come to the U.S. to continue her education after experiencing profound challenges and loss in the Lebanese Civil War. She earned her PhD in chemistry from Portland State University, and there discovered her love for dynamic proteins. We're looking forward to your talk, Elisar. Congratulations on this well-deserved honor.
It's not on? It is on. And thank you for coming. This is a really great honor, and I'm very privileged to receive it and very privileged to be talking to you today. So I showing here on the screen proteins from my lab. And these are proteins you don't see with a microscope, but you infer from a lot of data that you collect. And what you see among them is there is a shape, a certain shape here, and a lot of noodles or curly hair around it, shapes and noodles. Also here, a beautiful shape, and also a lot of disorder. And so this is the common theme. You have an order or shape and a lot of disorder around it. And so these disordered proteins, or intrinsically disordered proteins, have been underappreciated for decades, and my lab has been instrumental in bringing them to recognition. And I'll tell you some examples about that today. And because science doesn't happen in a vacuum, I also tell you how I managed to build a career and have a family, in the hope that my story will resonate with some of you or maybe inspire some of you. So this is one of the pictures from the Gilfillan family, and I want to welcome the Gilfillan family here. And this is a picture I like because it shows the family around the fireplace. I also have pictures of families, and watching my daughters grow from identifying rocks to climbing on rocks. See here, Mary's on this rock. And Wendy winning her first race in kindergarten to being a college D1 athlete, still racing, has been the outlet that I've so much needed during difficult times in this career. And I also want to acknowledge my parents and my brothers. So I grew up with 4 brothers, and growing up in 4 brothers helped me in not being afraid of having a voice around the table with all the men. So I grew up in Beirut, Lebanon, a small country. This is Beirut, and this is the United States. And in a multicultural family. My mother was from an old family in Beirut. My father was from Aleppo, from an Aramaic minority community who left Aleppo and came to Lebanon to pursue higher education. And Beirut is a beautiful coastal city, and my parents had careers in teaching, and they had kids, and this is me. And we're both teachers, and they instilled in us the love of learning and the love of teaching, actually. And in spite of the civil unrest in Lebanon, they had dreams for us, and we had dreams for ourselves to pursue education. And my father's motto was, or they instilled in us, that you really need hard work, and you really need to love what you do and be patient for a perfection. I went to the American University of Beirut. This is pictures from when I was there. These are pictures when I was there a couple of years ago with Wendy, the same places. And it's a beautiful campus. It's the chemistry department overlooking the Mediterranean. And I majored in chemistry. And the motto of the American University was that they may have life and have it more abundantly, because education So this motto was ingrained in me: education is the way to a better life. Now, even though this was a beautiful campus, I did not stay there all the time, mostly because, I'll show you why. So this is Beirut here. And when I was going to college, there was well, actually I should say that the Civil War spanned part of my childhood and teenage years and my adult life. And during the Civil War, there was a West Beirut and an East Beirut, and I was in East Beirut here. And to go to the university, it takes like 11 minutes. But when I was there, there was a green line. There's no man's zone, so you can't really cross. And the way to cross is to go around the city and walk for part of the time, because so you leave a car here, you walk, you get another car here to get to the campus. And I stayed on campus the first year, and then the second year they had an off-campus program that I transferred to. But because I was in chemistry and have physical chemistry and analytical chemistry, you couldn't do them in just a new building. So I was going a couple of days a week to do the labs. And while here people are living in peace, we would come just to go back from after a whole day of labs to my home. You couldn't get there because many days we couldn't get there because there was shelling, and many times it would be like the buildings next to my house would be on fire. So this was our daily life, and we made it through, and we lived through it, and I graduated. And around the same time, the American University president was assassinated. He was American, so they removed all the university professors who were American, go back to America. And so there were no graduate programs anymore. So I ended up teaching high school, and this is a picture of me in a chemistry lab doing all kinds of experiments. It was a fun experience. I discovered that I love teaching, and I really liked the experiential learning. But this wasn't what I really wanted. I yearned for more. But leaving Lebanon and going to the US was a very, very challenging financially. And I did not want to leave. Well, I guess I was scared to leave and in the unknown till my grandfather was killed by shrapnel the day before. And then I said, I'm not going to stay here to die. But it was a very difficult time war-wise, and there was no airport. So this is here, Lebanon. There is no airport. There was no port to leave by sea. But the way to leave was to take little boats, so fishing boats, to the middle of the ocean, and then transfer to a big boat to take you to Cyprus. And from Cyprus, I took a plane, one-way ticket to America. One-way ticket because we didn't have money for a two-way ticket, but it also was like I knew I wasn't going back, so I have to make it work. But what I had with me was a can-do attitude and extreme work ethics and adaptability and resourcefulness from my life in Lebanon. So my first stop was at Portland State, and I was a tourist. I was on a tourist visa, and I didn't have any official transcripts, but I went there and I talked with the chemistry department, and they took me, and there were no TAs left because it was in the summer, but they gave me a job in the stockroom to prepare reagents, enough to convince the INS to give me a visa. And then I made friends, I graduated, and my best accomplishment while I was at PSU was meeting my husband and my future husband, and made a life together. I graduated. I learned mainly how to do NMR on large proteins. This was at the early days of NMR on proteins. And as you see here, there's a lot of peaks that are very crowded. So to analyze those was impossible. What I was able to analyze is the differences. So you see the arrows. Oh, there's a new peak here that's not there. But important to remember, too, that I came to have a PhD in chemistry, but I never took biochemistry before. I never knew about proteins as an undergrad. So I did that in my PhD. And there I discovered that I really loved to be in the lab. The lab was a sense of belonging. I felt I belonged. I was so homesick all the time, and the lab was my safe place. So from Portland State, I went to Minnesota to do a postdoc in protein biophysics. And in biophysics, we ask big questions. Like our motto in biochemistry and biophysics is revealing how life works. So we really want to ask big questions, not just how to make things. And the big question that the lab was asking at the time, and a lot of other labs were asking, is how do proteins, so this is a protein, which is a chain of amino acids or a lot of atoms, doesn't matter, a lot of atoms in here that don't have a shape, but then they take a shape like this, and when they take this shape, they expose this green thing, and that green thing can come and fit into another protein, and that's how this protein works. So proteins are what does the work in the body, and they do the work by binding, by sticking with other proteins. But the question was how does this happen in a very short time? So you have this unfolded, disordered protein become folded in less than a millisecond, because there are so many ways for this protein to fold, but to get to the native conformation, native form, in a short time was a challenge that people were thinking about. So before I joined the lab, there were other, as I said, this was a big question in the protein community at the time, how do proteins fold? And Tom Creighton, who wrote the book about proteins, he came up with for this particular protein called BPTI, he came up with a folding pathway that you don't have to sample all conformations. You just have to go through many steps, and you get there. And this was accepted, but then a couple of years later, another researcher at MIT, Peter Kim, he said, what Tom Creighton said was wrong. There is this other way. And so there was this big argument in print. The subject is too hot for protein chemists to touch, and everybody's arguing about which one is correct. So in the meantime, the lab before I joined, they were using the same protein as a model to understand how proteins fold. But contrary to what Kim and Creighton were doing, they wanted to remove these yellow things here. These are like bridges, disulfide bridges. They removed these disulfide bridges and kept only one. And the only way to make a protein like that was to do peptide synthesis, which is one amino acid at a time. So they made it, and the student who made it didn't want to deal more with characterizing it, because the idea is you make it, and you want to see where are the regions of the protein that fold. So you have a protein in a test tube, and you want to know what parts of it fold. And that was something I was really good at. So this is when I joined the lab, and these are the kind of spectra that we collected, and a lot of other experiments. And I want to spend time here to explain. So these are NMR spectra, and what you're looking is at peaks. So this is the same protein, but you see these spectra look different. This one is a folded protein, has a beautiful shape, and you see those peaks are spread out. While this one is an unfolded protein, the same protein, but then all the peaks are clustered together. And this one is partially folded, and the peaks are a little bit spread and also very broad. So this is a very easy spectrum to get, and people have done this and published the structure. These are much more difficult because, as you see, the peaks are on top of each other. So this is the work I did, and we came up with this. We want to understand how a shape like that becomes this native form that does the job in the cell. Okay, so that was the question, and we came up with a pathway that says, all right, you do one disulfide, you force folding to happen farther from the disulfides, and there is a lot of other conformations, but all of them have folding in the core of the protein, in the part of the protein that's buried, and that was the idea. Is that instead it doesn't matter. So what we said then, all these arguments about the disulfides bridging and which disulfide happens first, doesn't matter. Because any disulfide would do the same thing. You need only one to have folding in the core of the protein. So this was the big story, and I got several papers out of it, and got invited to give talks and won an award. And this is the lab of Claire Woodward, and after 4.5 years, it was time for me to establish my own lab, and this is when I got my first job, but before that, we had a little complication here that so I had to start a lab from scratch with a 3-month-old baby, but more importantly, I had to think of a project that's not BPTI, that's not a model proteins that everybody's working on. We wanted to think about some other proteins that's interesting before I go on to my other lab. And so the lab across the street we're working on a protein called dynein. So dynein is a motor protein. So what that means is that it walks along microtubules or like train tracks in the cell and carries all this big stuff with it. So it's a protein that walks and carries stuff with it. And when it reaches where it's supposed to reach, it drops it. So I was very intrigued by that because I was trained to think about how proteins fold, what segments come together. And now this is a large complex made of a lot of different proteins. The question is, how do they get together? So this was a really cool question to ask. The one problem is that it's the scale of the jump is too big. This is BPTI that I used to work on, and this is the big protein that I want to start my lab on. So that was a very ambitious, but I was interested in the functional relevance of this protein. It's related to chromosome segregation, et cetera. It can be involved in a lot of medical effects. Okay, so at the time, this is when I was starting to think about starting my lab. This was the time when the eukaryotic genomes have starting to be sequenced. So before you sequence one protein at a time, now the whole genome is being sequenced. There is that resulted in if you know the sequence of the DNA sequence, you can know what the protein sequence is, and then you can start figuring out what these proteins do. And so there is this explosive growth in bioinformatics and computational biology. A lot of studies using NMR to see how you can get structures with NMR. And structural genomics, which the idea behind it is you have all these sequences of proteins, we don't know what they do because they are all new. And the way to know what they do is to get their structure, and maybe structure will tell us. The structure means the shape. The shape will tell us what they are doing. The problem is I didn't know any of this. I didn't know what to do anything of this. I didn't have any cloning experience. I didn't have any protein expression or labeling experience. So here I'm starting a lab, a very ambitious project, and with new learning as I went along. So I want to take a little minute here to emphasize people have already always thought about structures from the time of Linus Pauling. Many Nobel laureates for solving structures. There's the structure of hemoglobin, structure of insulin by Dorothy Hodgkins. Christian Anfinsen stated that the proteins have in their sequence the ability to fold NMR structures, the structure of the ribosome, the cryo-EM revolution, and then most recently this year using artificial intelligence to determine structures from sequences. So all of these techniques are to get you the structure of the folded region. Remember, my talk was beyond the fold, and so we'll get to that. And the reason people cared about structures is because structures are pretty. They tell you what the proteins do. I mean, this is a very pretty structure that has a big cavity here. You can design a drug and put it in there. So it's a very important thing to know for drug design. And so the structures are important. People spend a lot of time about them. And I'm starting my lab. You know, this is a protein nobody knows anything about. There's no biophysics done on this protein. We don't know what it's doing. Let's get its structure. That was a very good idea at the time. So while I was still a postdoc, this paper came out in Science that says there is this protein inhibitor of nitric oxide synthase. And it turned out that same protein is the protein that it was my baby that I was building my new career on. That's called LC8. And I was happy that it had relevance for nitric oxide published in Science. It means it's important, but that meant other people are going to look at it as well before me. And that's what happened. So while I'm still starting my lab, there are these papers that solve the structure, and it says it's a monomer trimer, and this other paper said it's a monomer, it's an NMR structure. And I stopped reading the literature after that because I was so scared. And then because we knew the monomer is wrong, because our experiments were telling us it's a dimer. Dimer means two pieces coming together. So I said we still have a story here, till the structure came out for the dimer. But we still had the story because in that paper they said it was at high pH, and actually with our results that came out 3 years later, as it was my first paper from my first, and those of you who are starting labs know how difficult this is, to just getting scooped and then publish a first paper after 3 years that explained that that it's not a monomer trimer, it's a monomer dimer, and the pH, and this is how it looks like, and we explained why it dissociates at low pH, etc. And I should stop here to say that the first paper from my lab was done by undergrads. It's these two undergrads here, Brian and Dan. Dan was the first, this is a recent picture. I reconnected with Dan last summer in Chicago. But he was the first student in my lab, and he was helped me open boxes. And Brian was I was preparing the proteins for them so that they do the analysis. So undergrads in my lab would think this is hilarious. I'm making the proteins, the students doing the analysis. And then I'm showing here pictures of more recent students who are pursuing PhDs. Have Coben, he's getting a PhD at Duke, Delaney at Stanford, Sanjana in London, etc. So I've had a very good luck with undergrads in the lab, and I wanted to give them credit. Okay, so the structure is done. What do we do with the structure? So the story doesn't stop with when the structure is done. So remember, this is the big complex we want to look at, and we wanted to look at its interaction with this large protein. So this is a 640 amino acid protein. It's considered a large protein. And there was domain analysis. We could do sequence analysis at that time. So we could see this is a different fold. This is a fold that's different from this. And we were interested in seeing where it binds the other proteins in the complex. And so my student made these two different pieces and found out where they bind. And they said they bind here where it's colored. But before we published that paper, we also had 2 other papers, one for each light chain that were published. And then I decided I want to focus on making the story about this new thing that we have never seen before. So you have here a long chain, a long piece that has no structure. So this was very unusual. So it's unfolded on its own. You didn't have to do anything to describe that, to open that structure. And it's also, what was really cool, is that this is the part that's functional. This is where it's binding its partners. So this is the first evidence that disordered regions were important. They were the longest, and they were the longest characterized experimentally. And then even after binding the partners, they remained disordered. So all these were new, nobody knew about, not just with dining, just in the field. So this was at the time when the people have started to think about, okay, there are proteins that are unfolded on their own. And then later, after I moved to Oregon State in collaboration with Andy Karples, my student Justin, solved the structures for these. So we can solve structures, and they look very pretty. But what's important is that the surrounding sequences are disordered. And the question is, what is the disorder doing? So as I started saying, there are proteins that have to fold, take a shape like this, and be like a lock and key when it binds its partners. And then there are proteins that don't really fold. They have a lot of disorder staying in them. And there is proteins that are unfolded, but then they take a shape, like you see here, when they bind their partner. And our proteins, or the set of proteins that I will show you, are more like they are disordered, and they bind this LCA dimer. And they remain very disordered, except at regions where binding is, and at regions somewhere in the protein. So NMR is the only method to look at disordered proteins. And you would think it's the only method, means it's the easy method. It's not. This is LCA when it's a folded protein. Look how beautiful these peaks are. There's a big spread. You can put a number on all of these peaks. But then you look at this should have more peaks, but look how on top of each other, these peaks are. So this is a very challenging protein to work with or to look at its NMR. But I have two heroes in the lab who helped me start out my lab. Moses was the first to make the intermediate chain, the disordered region, and get it to work. And Aoife was the first person to do NMR on it. Fast forward 20 years, we know now one of the reasons that this protein is disordered is that if it is a folded region and a lot of disorder around, and that the disorder is used to cover the active, the part that needs to be there to function. This part here, where the yellow is covering the gray, this is the part that functions and now it's covered. But then when you add proteins to it, you open it up, becomes like this in this cartoon. So now it's opened up and it can bind other partners. So that's important for proteins, that they don't need to function all the time. They are inhibited, and when they are needed, and they're inhibited by these disordered tails, and when they are needed, they open up and do their work. And that's work of, we were able to do this because of the extreme talent of Kayla Jara and Yujan, who was able to make this large protein. Okay, so an offshoot of dynein is this little protein that we started working on with, which is called LC8. So we got the structure, and it binds partners in the green. So the partners that get together with it are in the green. You see there are two. So there are a lot of proteins that bind LC8, and the idea in the dynein field is shown in this cartoon, is that dynein is a motor protein. It goes on train tracks or on the street. And it has this light chain, LC8. So it can bind dynein and connect it to a lot of other things. And that's what's called adapter. It needs to be connected to other things. So it uses LC8 on one side to bind dynein, and on another side to connect it. And this was the model accepted in the literature. Every protein that gets discovered to bind LC8 shows a model like this. But we have shown that LC8 binds two dyneins. So if it's binding two dyneins, there's no place for cargo to bind. So we proposed I spent a whole summer trying to figure out, to write a proposal, and we wrote a proposal, but then I said I'd write a paper about it, and the paper is more like a concept. This is a new concept. What we think is that LC8 is made in dynein, or it's discovered first in dynein, but also exists in other systems. So, and these systems are different colors, so it can bind, it's like a date hub. It can bind and go to the same dance with a different night or different place with its partners, but it's doing the same thing, the same dance. And what it's doing, it's taking disordered proteins, bringing them together so that they do something else. So you see there are these two disordered proteins come together with LC8, and that's what it's doing in the cell, and that's why it is important. So this was a big paradigm shift, because everybody in the field was thinking it's a cargo adapter, and now we tell them it's actually a hub, means it's interacting with everything. And I wasn't part of the dynein community, so I never went to their meetings, but then I was invited to go give a talk in a dynein meeting, and then people would come to me and say, oh, so you are the Elisar Barbar who came up with this idea, and now we know. So once they see me, they know how someone can come up with this idea. And that's because I look different. I come from a different place. And so that underscores the importance of like bringing people from different cultures or different upbringing because they think also differently. So it's not just to fill a quota, it's also we advance science this way. So this is so LC8 is a hub, binds more than 100 plus partners, and they're involved in a lot of these processes. And these are examples of what they do. So if it brings these two together, then you have different phenotype here. It's involved in RNA, keeping RNA together. You can also have several LC8s in a row. I'm going to tell a story here about another a protein that binds LC8 that helps. That's removing it affects the lung development. So this is a lung here. Without that protein, the lung is smaller. And this is Drosophila wing. Without that protein, Drosophila has a smaller wing. But then you add LC8, even though it's not that protein, it's called the knockout is ASCIS. You add LC8, you regenerate that, you make that lung bigger again. So I knew about this from Jörg Heierhorst, who I met in Japan at a meeting. He saw my name on the roster of speakers, and he came just to talk to me, so was very flattered. And he wanted to convince me to work on this. This is the protein, it has a DNA binding domain and has this big tail that's disordered, but this is where it binds LC8. And they wanted to know, what is LC8 doing? Why do we have this many LC8s? And I came back to the lab, and I was lucky to have Sarah Clark in my lab, just starting, who had no fear of doing anything hard. And so she, after several years of working on this, she came up with this model that this protein, it produces LC8. When there is a little bit of LC8, it produces LC8. Then LC8 comes back and binds. So this is all full of LC8. And it stops producing. So if you have a lot of LC8, you stop production. If you don't have any LC8, you start production. But then the majority of the protein is in this region here, where it is partially sensitive to the concentration. So if you need more LC8, you give you more LC8. If you have a lot, we stop. So this is more like a rheostat. So rather than an on and off switch between transcription on and off, it's actually you just tune it to the amount you need it. Another example that I also met at a meeting is a protein called KANG. This is with Anna Akhmanova. And she said, oh, we have this protein that binds LC8, and we don't know where it binds. And it looks like there is one binding site, but we don't know where. And so we looked in this region that's disordered. So this is a whole 600 amino acids that have no structure at all. But it turns out this is where LC8 binds, and it binds LC8s in like a rod. So you have several LC8s in a rod, and this is more like an on and off switch. So this is off and this is on. You don't have anything in between. You remove LC8, the protein becomes disordered, so it cannot do what or disordered protein would do. And Douglas Walker, postdoc in the lab, showed that actually, if protein does on and off, or it does tuning, it is encoded in the sequence. So in the protein sequence, you can predict from the protein sequence what kind of a complex you get, which was very revolutionary for us, because now we can make any proteins we want. If we want them rods, we can make them rods. If we want them flexible, we can make them flexible. So another example from of LC8 partners is in viruses. So rabies virus has 5 proteins. One of them is a protein that binds LC8. And Nathan Jesperson did the NMR and all the studies on it and showed that this is the model. You have this disordered linker here. This is the part that's important for binding. But these are these disordered linkers that makes the protein move in all kinds of directions. But then when LC8 comes, it stops it from moving in all these directions. And now the function can happen. And this is when rabies when mice are infected with rabies, if there is LC8, the mice die. If there is no LC8, the mice live. So this is the active form. So you need to be restricted rather than flexible. Now, when we published this, that was just at the beginning of the COVID pandemic, and we were thinking of viruses. So then we said, okay, what similarities we have between rabies virus and coronavirus? And Heather Forsyth in the lab showed that there is quite a bit of similarities, and we were able to work on this protein and show it's a dimer with a lot of disorder. And what this protein does, it covers it. It binds RNA and covers it, packages it, covers it all in a small space, so that this is the picture of the coronavirus that you're used to seeing. And that's because this protein is binding that RNA, and it binds as a dimer. And Heather won the Dance Your PhD competition on this work. And Hannah Stube is completing this work. Here we have viral packaging. This is the virus before it enters the cell. This is the virus after it enters the cell. The RNA moves out, it expands. And the way it expands is because you have this disordered linker that can open up. And when the disordered linker open up, we have we have extended the RNA. So without having to change where RNA binds, by just changing the flexibility of these linkers, you can make the RNA more open and be the switch between viral packaging and replication. So there are, we don't want to forget the lessons we learned 30 years ago. Some of them are still applicable. This is back to the BPTI story. What we showed then is that you put a cross-link to bring two chains together in the same protein, and you force folding. While here, what we're seeing now is you cross-link with LC8, which is a dimer, bringing two chains of two different proteins, or two proteins of the same kind, but they are different proteins, and then you force folding. Another lesson we learned is that partially folded proteins can be multiple conformations and have the core primarily folded. In the disordered proteins, you don't have a core because these proteins don't have a core. A core means the inside of the protein around which the rest of the protein binds. And here, you have multiple conformations or multiple shapes, but you have one shape that ends up being the binding to a partner. So to summarize what I just told you, disordered regions or beyond the fold dictate function. The function could be here, but if you're covering it by disorder, it's not going to function. It only will function when you open it up. In this case, the function or the role or whatever it has to do is here, but the disorder here tells it when it does its function. So even though the protein structure is where the function happens, the disordered regions outside of it is what tells it when to do it and how. And we've discovered a new class of proteins that are calling IDP duplex scaffold. They come in many shapes. You can have 5 in a row, you can have only 1, and you can have several, and you have 8. And that could be either rods or tuning or multiple different shapes. So the impact of this is that LC8 binds more than 100 different proteins, and they could be in any of these shapes, and we have the power now to figure out which ones are the ones that form a rod and which ones that form just a flexible region. So the impact of my work is we kind of brought to the front the importance of disordered proteins that were underappreciated for decades, and also in starting a new field, the field of LC8 and whatever it does with its partners. But the real impact, in my opinion, is in the people we mentored along the way. So I've had the privilege of working with all kinds of training people at different levels, and high school students, high school teachers, undergraduate students that have gone on to win awards at national and university awards, and graduate students winning awards nationally and university, and so postdocs. And so I was very lucky to have been able to be one of this, to bring this about. And these are some pictures that I'll show you. So this is an older picture. This here, Andy Karplus, long-term collaboration. Yujwan was a research assistant in my lab at the time. I'm very glad to see that she's back. Ifwa was a postdoc, and she's here as an associate professor. Rick is also here as an associate professor. Let's see who else. So, and then we had a reunion when Justin won the College of Science Young Alumni Award, and had former and current labs. I will scroll these pictures fast, but what the thing that I want to show you is there are a lot of them, and they are diverse, and they are happy. So that's and this is the names I listed here are members of my current lab, and I'm very lucky to have worked with excellent people to get here. None of this would have happened without funding. And I'm listing here all the funding to emphasize how important funding is, especially now with the attack on funding. And remember that the United States produces more influential health science research than the next 10 countries combined. So this, there is federal funding, there's also foundation funding, and also in every NIH dollar goes back to the economy, because this money is used to pay for people, it's used to pay for reagents, it's used to pay for infrastructure. And one thing really important is for people to know is that NIH doesn't give out money. We work very hard to get this. For every number you see here, there has been like 10 that were not funded. And every proposal has a story, and every proposal is written in blood. And this is one example. So the example that brought the NMR, the funding that brought the NMR, was a proposal I worked on for 10 years before we got the NMR. And getting the NMR was incredibly influential, because we got Patrick Reardon as director, that he opened the NMR to the whole community, not just at OSU, but also to the Oregon universities. I've been lucky to have my science take me to all these places. After Ohio University, I made Oregon State University my home. I've been here 21 years, and throughout these, I did excursions to New Zealand, and spent a year at EMBL and Heidelberg, and most recently as a visiting professor in Munich. But there's no place like home. So I've had a very supportive community at Oregon State, and so there are people who have retired, and I see some of them here. There's Joe and Andy, and this is the new group. So it's a great place to grow and to get into leadership, and a great place to have a community. And I want to thank my family. I've made many mistakes starting out, and it is a challenge for women to have it is still a challenge for women to have a career in science and raise a family. But I'm assured by my daughters that they had a good life, so I wasn't very. And of course my husband, and it takes a village to get all the help you can. And in honor of our recognition of Women History Month, I want to honor my former postdoc advisor who introduced me to protein dynamics and was a major supporter throughout the years, and also to my mother-in-law, Mike's mother, and my mother for doing a lot of the taking care of kids, and even living with us to help us when we couldn't have daycare, and traveling with me on meetings, and being a role model for what women can do and raise a big family and have a career. And I will end by dedicating this talk to my dad, who passed away early this fall, for being a role model in his passion and enthusiasm and zest for life, and also for reminding me that our legacy is in the people we mentor, and the opportunities we give, and the lives we change. And take-home message is we are the stories we carry. I mean, I'm like this because I have a story, and I'm sure your stories will make you the people you are, and how we respond to crises, and how we set expectations of ourselves and others, and how we face challenges. And science, I've mentioned that led lots of collaborations internationally. It's important to remember that science is not confined by international borders, and especially in times we live in right now. And for me, doing science wasn't a job, it was a way of life. And it changes life, it changed my life, and I hope it changed the lives of people who worked with me. And I will end by a call to action that, considering that the attack on science and academia and education and research, we need to respond to these direct attacks, and so that we can keep this magical place, and I quote here, that's shown as a beacon of freedom and opportunity to so many who had come before. So thank you, and I'll be happy to answer questions. Thank you.
We have time for a few questions. I'll let you just take them out.
Okay, yes. There are microphones if you want to go to the microphones, and be happy to answer questions.
It sounded like Oregon was the first place you came to in the United States, but how did you choose Oregon?
Yes, I missed some of the details. So my brother was going to school here, and I was on a tourist visa to go visit him at one time, and I chose that. And then, but I had no connections with Portland State, and I made the connections after I came. Abraham has a question.
Was there ever a time in your career that you thought you don't want to do this anymore? And how did you overcome that?
There were lots of times. I had a very hard starting time. As I said, I chose a very ambitious project. I had a 3-month-old baby. I was in the lab. I was in an organic chemistry department. They wanted me to teach at 8 a.m. I hope this is, we can remove that from the recording. So I talked about research, but I didn't tell you about the teaching part that was also very challenging for somebody starting out. And so being scooped one time after another, especially the second time, I had, those papers came out while I was ready to submit our paper. And I submitted our paper anyway, because we've done the work and we did the different methods, but we came to the same conclusions. And the reviewers sat on it for 2 months without telling us what they want. And I had a proposal that is under review. And then the paper was rejected. The proposal was rejected. I could not get out of bed after that. And I remember it's very vivid in my memory. I went and mowed the lawn when I got out. I never mowed the lawn again. But so when, well, how do you recover? It is important. I mean, that's why women leave science early. It's just tough. And recover success. Success has really helped. I mean, my best recollection is when I got a phone call to tell me my NSF career was awarded. I remember it very vividly. It's getting success and being rewarded. So it keeps you going. And then seeing people telling you how much they loved working with you, getting those cards. So write those cards, because when you get them, it changed my life, and I cherish those a lot.
Thank you. So you've worked on quite a number of projects, and you've been to quite a number of places. My question is, what project was your favorite, and what place also did you like the most in your career?
Oh, that's a good question. I do think I'm partial to LC8 because that's something I brought to life. Anything that with LC8 is, the hub idea was a really clever idea, even though by my standards. And it really changed, made a big impact. Every paper that comes out reference our paper. And so I think I'm very partial to that. The coronavirus project was good because it made me feel that we're doing something constructive, like we're in lockdown, can't do anything, but actually we can. So this was also a good feeling.
How did you manage just being a woman so high in your career, and just like how do you manage with internal feelings of maybe imposter syndrome, especially, you've achieved so much, and definitely I know in my own experiences as wanting to do science my whole life, I've always felt this sense of it's, I didn't do enough, I never did enough, so how did you manage that throughout your career?
So I grew up in Lebanon, and parents emphasized math and science. I didn't have an imposter syndrome. That's not true. I hid it so well. I said I came with a lot of confidence. I projected confidence. I went to Portland State, and I talked with the professors, and they accepted me by just talking to me. I projected a lot of confidence. And that doesn't mean that how I felt it, but I had to struggle to not to struggle. I mean, I knew this was my only way to succeed, and I wasn't scared of, I wasn't apologetic. I'm never apologetic about standing up and providing, like, showing off, not showing off, showing up.
Thank you again. Earlier on, you mentioned getting scooped, like you are working on a project and then another group is still working on the same thing, and then they might get it published before you, so, which is very discouraging. So my question is, how do you get that to not happen to you? How do you get your work out there quickly so that no one else publishes before you?
Well, that's a very good question, actually, and I wanted to say that and I forgot. So, we were going for the low-hanging fruits. Okay, there's a structure, and everybody was going for that. And then when the lab that scooped us for the interactions, they looked at short peptides. We were looking at the big protein. We were very ambitious. Looked at the big proteins. They did the short peptides. But then we discovered that we are so good with the big proteins. And this is how we made it. It's very difficult to do. Very few people will do it. So we did it. And then we were not scooped. I don't think we ever got scooped when we were working on the big stuff. So you want to find what you're good at, and you don't want to do what everybody else is doing. And make what you're good at better. You really dig in and get better. We were doing physical chemistry, thermodynamics on large stuff. Other people were doing it on this. So we were not scooped because we were doing hard stuff.
So how did you determine that the proteins were actually loose, unfolded? You can't see them.
You can't see them.
So how do you know?
Oh, I skipped that. Skipped all of it. So there are a lot. We're biophysicists. We use a lot of instruments and techniques, and there are a lot of techniques that will tell you. But one very simple thing that the very first thing we did was, if a protein is disordered, if you chop it by enzymes that chop proteins, it can chop very easily because it's open. For proteins that are closed, they don't chop. So you can look at the gel and see, I have several bands here, or I lost my protein. That was very discouraging because they will be degraded, that's the word we use, on their own. So you make the protein, 2 days later it's gone. So that's how we discovered it the hard way first. And then we figured out how to make it stay longer to do experiments on it. Yeah, I think working on disordered proteins was a happy thing because I kind of like to say that I brought them to existence, and our lab was major in the field to make them important. But they also rescued me. They rescued my career because everything else I was doing, I was getting beaten. And then when we got to the disorder, was a
Hey, thanks, Elisar. You've got this hub that folds all these proteins, and where is it? And is there a hub of hubs? I'm imagining that there's many of these, and so.
Yeah, so by definition, a hub protein is a protein that interacts with a lot of other proteins. So it used to be like one protein, one interaction. That was the thinking. And now one protein, a lot of different interactions. There are very many other hub proteins, okay? So 1433 comes to mind, calmodulin is another. Any protein that binds a lot of partners is considered a hub. We, of course, think LC8 is unique because it does something. It just doesn't interact only. It brings these two chains of disorder, brings them together and makes that aligned, either ladder or rod or string. Okay, so I don't know if any protein like it. Yeah.
We could do maybe one more question.
Oh.
Thank you. I was wondering, did you come up with any general principles which could tell you whether a protein sequence is going to be disordered?
Yes. So, so when we got started this field, I would say, started around 2000, when there was a lot of bioinformaticians, and all what they were doing was looking at the protein sequence and say, is it disordered or not? So there are a lot of programs out there. So the field was governed by bioinformaticians. There were no experimentalists. And what we brought was the experiment. We took this sequence that they said it's disordered, and we made it, and indeed it was disordered. So there are a lot of programs out there that you can, like now if you're working with new proteins, the first thing you want to do is do some sequence prediction. What structure do you get? And then you do the experiment, because that guides the experiment. But what we brought to the field was the experiment.